Cell Paper Flicks presents Nanotechnology Meets Mechanobiology. The ability of an organism to interact with and respond to its environment is an essential component of life. Cells interact with their environment through chemical ligands, with each other through ligand-receptor interactions, as well as with the extracellular matrix. Cell-cell and cell matrix interactions are often mediated by mechanoreceptors, receptors that propagate signal in response to force. Mechanosignaling is vital in normal tissue functions, such as cell migration, tissue homeostasis, and developmental processes. Diseases such as cancer often have misregulated mechanosignaling.
Signal transduction by these mechanoreceptors is extremely complicated but very well choreographed. As nanoscientists, we wondered if we could design an artificial system, so-called mechanogenetic tool, that manipulates activity of specific mechanoreceptors at any desired space and time. We realized such a system by combining nanotechnology with genetically engineered targeting of mechanoreceptors.
Traditional tools in the study of mechanosignaling include atomic force microscopy, optical tweezers, and magnetic microbeads. These technologies are large in comparison to receptors and engage many targets. They restrict receptor movement and perturb the mechanical environment. This results in nonspecific receptor activation, even in the absence of the external force used to promote activation. Nanoparticles are of a similar size to receptors and do not perturb normal receptor function. We collaborated with the Cheon lab at Yonsei University in South Korea to synthesize magnetic plasmonic nanoparticles.
The chemical design of plasmonic magneto-nanoprobes is critical. It should be as small as the size of typical receptors and has the multifunctional capabilities for optical imaging and magnetic force generator.
The nanoparticles consist of three components: a magnetic core for force generation, a gold shell for imaging, and a targeting region. Nanoprobes that integrate all three functional components are slightly larger or comparable to target receptor sizes. Using the targeting region, nanoprobes are specifically attached to target receptors at the cell surface, then manipulated with a magnetic tweezer. Spatial manipulation is imaged with dark field microscopy, and stronger forces can be applied to induce receptor activation.
These nanoprobes are extremely powerful and widely applicable. Any receptor of interest can be specifically targeted through the modular functionalization domain of the nanoparticles and then mechanically activated.
We decided to demonstrate the utility of these nanoprobes in two model systems. First, the cell-cell communication receptor, Notch, and second, the adhesion receptor, E-cadherin. Both receptors are known to be mechanosensitive, but the exact mechanisms of activation have been difficult to study. Notch and E-cadherin allow cells to communicate with each other transcriptionally and mechanically. Ligand-receptor interactions, clustering, and mechanical force may all be important in mechanosensitive receptor signaling. Using this mechanogenetic toolkit, we tested each of these interactions in Notch signaling and found that only force was required to activate Notch signaling. Additionally, we imaged actin recruitment to E-cadherin clusters and found that while clustering recruited actin, force was required for filament stabilization. Another exciting application of this technique is single-cell control of gene transcription. Using genetic engineering, any gene can be controlled through the application of force. Here we have specifically activated gene transcription in one cell in a field of cells. Activation can also be temporally controlled so that each cell turns on sequentially.
I think what excites me most about this technology are its applications in the studying of the spatial regulation of cellular signaling events. I say that because there's really an accumulating body of evidence that there's a tight coupling between the spatial organization of various cell surface receptors and the nature of the downstream signaling events that they activate. And this particular technology is really ideally poised for investigating that coupling particularly in the context of mechanical signaling between cells or between cells and the surrounding environments.
And this is a really big development for biologists who would like to understand how the systems that they are investigating really work. Many things that previously were more difficult to do, nanoparticles enable that to happen in a much more systematic way. So I think that represents a very big development that's quite exciting.
I believe the most important components enabling this work was collaboration between scientists from different fields, including nanoscience, chemistry, cell biology, and microscopy.